Hey, hello everyone. My name is Bingxun Cai, and my advisor is Professor Yu Wu. Today I'm going to talk about using fractal dimension analysis with the Desikan Atlas to detect the brain age of the healthy adult. And this is introduction. Normal aging is an important issue for medical study. The rate of changes in the cortical regions is considered a predictor of aging effects. The brain atrophy rate can be a predictor of aging. There are several methods to evaluate brain atrophy, which in my research uses fractal dimension. Fractal dimension has been proven a better measure for atrophy detection. But regional atrophy during lifespan is less explored, so I isolate the brain regions and calculate their FD value. Desikan Atlas is somewhat like a Google map of the brain, and is used in this research. Then compare the gender and the different stage of adulthood difference in brain atrophy. And now we will see using fractal dimension to predict brain age is less valuable or not. And this is materials. A total of 708 healthy adults participated in my research. There were 259 women and 449 men. All the subjects were divided into several groups according to their ages. This is proposed. Thickness of cortical regions is related with brain age. So using MRI data of healthy adults to compute FD of every age, then using FD to predict brain age. And this is data process. First, collect the raw image of every participant. They are T1 weighted images. Then the raw image was normalized into a volumetric template and segmented into gray matter, white matter, and cerebrospinal fluid. In the third step, the cortex of the gray matter was pixelated and aligned into 68 distinct regions of interest in the Desikan Atlas. Last, measure the FD value of each pixelated region and the lobes for each participant. A recon O is the main step of free surfer. It can be separated into three parts. First part, process step 1 to 5. Second part, process step 6 to 23. And third part, process 24 to 31. This table is 68 subregions in the Desikan Atlas. There are 5 mainly regions including frontal, limbic, temporal, parietal, and occipital. Odd and even numbers are for the left and right. This research used the box counting method to evaluate the complexity of the human brain. The value of the FD was obtained from the slope and the fitted line. A more complex cerebrum has a higher FD value. And this is FD calculation and integration. After free service, the FD calculation will be the next step. The FD will be calculated by using MATLAB code. There are 86 FD of the region to be calculated, but not all the regions are used. There are several FD. I will remove them. Then, integration all the data into Excel. The Excel includes FD, age, and the case number. And this is brain age predict. I will input all the data and separated them into 80% training group and 20% testing group. The training group will find out what p-value of the region is less than 0.05, and then they will be used to compare with the testing group. The testing group will predict the age of the brain and compare the predicted age and the real age. This result is a mean standard deviation of predicted age, and the picture is distribution of the predicted and the real age. And this is the result. These are my result of brain age prediction. They are divided into 3 groups. All participants, male and female. The first time I predicted the age range from 30 to 85 years old participant. We can see that the male group got a better result because there are more data than the female group of training. But the result of 30 and 85 years old groups are not really good. Because the data distribution is not linear and uniform, and I try to take out the data of age range from 30 to 40 and 65 to 85. The result of are better than the 35 to 85 years old and 30 to 65 years groups, but it still need to predict those 2 ranges of age people. So I have another idea to solve the problem and the data uneven distribution. This is the data distribution picture. We can see that at 30 and 35, 36 to 40, 71 to 75, 76 to 80, and 81 to 85 group. Data are not as much as other groups. That makes predict will not accurate. So I am trying to use the sliding window method. For example, I take our 41 to 45 years group to explain. If I choose that the window length is 3-year range, I will randomly take 10 data from the 41 to 43, 42 to 44, 43 to 45 years group. Then the data distribution will be more linear and uniform, so the training result will be better than the origin. This is my another idea. If I can compare every 5-year group of their FD to find out differences in their FD value at specific regions, then I can predict the brain age at what group of the year. And that might be a great method to predict. Okay. And now I will focus on 2 points. First, because the frontal and the temporal lobe are 2 mainly atrophied regions of normal aging, so I will focus on these 2 regions to find the relevance of age and the FD value. Second, I will use the sliding window method to make data distribution more linear and uniform, and hope I can make the result mean age error less than 5 years.